Thanks, everyone. Can everyone hear me well in the back? Great, cool. So yeah, it's really great to be here. I'm Shams, and in this presentation, I will talk about my work at University of Maryland with Pratyush Tiwari, and I will discuss how we are using molecular dynamics simulations with machine learning, and we are trying to understand drug-target interaction mechanisms and calculate relevant free energies and kinetic properties. So before I start, I would like to thank Michael Schertz for introducing the very nice concept of Taco Bell formulation of statistical mechanics, and also a few other speakers, because I won't have to cover some of the ideas that they already covered. Okay. So in general, in our group, we look at problems that are involved in human health, and we focus on structure and dynamics. In our group, we have developed methods for calculating free energies out of molecular dynamics simulations. But in this talk, I will focus on the core principle that we developed there and that we are now extending to get kinetics for both protein and RNA targets. And specifically for systems in which it has been shown that kinetics is more relevant in efficacy compared to affinity. So I'm very excited to talk about the applications, but I'll also go briefly over the framework or how it works. And if time permits, I will also go over how we are exactly getting or extracting the interaction mechanisms out of the molecular dynamics simulations. And this also relates to transparency, or how we can comment on the trustworthiness of our models. So because we're interested in interaction mechanisms and kinetics, all of the molecular dynamics simulations is a natural choice for us, where we define interactions in terms of potential energy function, and then we integrate Newton's equations of motion to propagate the dynamics. But as we all know, that molecular dynamics suffers from the timescale limitation, where it's very difficult to sample rare events of interest. So there are a few solutions to address this problem, and we focus on enhanced sampling or accelerated molecular dynamics to solve this. So there are many variants of enhanced molecular dynamics that helps with the timescale problem with the different pros and cons. But a lot of them require something called the reaction coordinate. So here I'm showing the example of metadynamics, which we use in our group. And you can see the difference between normal molecular dynamics and metadynamics, where we deposit history-dependent energy to overcome free energy barrier and help the system to explore the free energy landscape. But there's a problem, which is how do we know how to deposit the energy correctly? So here, it's important to remind ourselves that statistical mechanics will actually not teach us useful representations. And it's up to us to choose what's the best representation for our reaction coordinate. And if we choose well, we will be able to get useful information out of it. So here we take inspiration from the idea of information bottleneck in information theory, which takes high-dimensional data as input. And if we tell it what we are interested in learning about as the output, it promises to tell us that it will identify which information is relevant in the input X for predicting Y. So it generates an information-rich low-dimensional embedding chi, as you can see in the schematic, by optimizing a loss function L that is defined in terms of mutual information. Now, this is typically implemented through a variational autoencoder. But again, there is also a problem. So in information bottleneck, we need to know what we are interested in learning about in the output. So typically, we don't know that. And for biomolecular systems, this is related to the metastable state that we are interested in. So here we propose our method, which is metastability-driven representation learning. And our method is called state predictive information bottleneck, where we recognize that biomolecules spend time in different metastable states, and their state-to-state transition is more important. Here we impose a mathematical principle that if a system is in a metastable state, it has the highest probability in staying in that state after a small time lag. And this allows us to formulate this problem as a self-supervised learning algorithm. This is a special kind of dimensionality reduction technique that treats data as time series, and it allows us to learn the low-dimensional space that we can use to bias metadynamics, but also it gives us we learn iteratively the output y, which tells us the metastable state definitions. And we can use this framework and extend it to infrequent metadynamics by simply using the theory that was developed more than 10 years ago, where we can run biased sampling, estimate biased residence time, and we can get back our unbiased estimate of residence time. So this is our overall framework. I'm showing an example of drug permeation problem where we pass a trajectory, train our machine learning model, and do enhanced sampling and iterate if necessary. So going to the application, so our first, for application, I'd like to highlight our very recent paper where Sum and Lee in our lab worked on looking at different pairs of proteins and ligands and identifying or calculating dissociation kinetics where the residence time spans across 12 orders of magnitude. So here, one interesting example is the Abel kinase and imatinib system, which is very interesting. And we actually looked at different mutants of Abel kinase and looked at how the dissociation differs between them. Some of the other systems that we looked at involved T4 lysozyme and benzene, FKBP protein, and dissociation of some fragments. So this is a summary, a representative dissociation, some of the representative dissociation trajectories, and this is the summary result where we can compare our obtained result with what has been known from experimental or computational studies, and we can see that the method does pretty well. Because we're using the theory of infrequent metadynamics, we can also get associated error in terms of the p-values where we also do reasonable. So here I'd like to emphasize one point, which is that our framework is very flexible and you can substitute different parts as necessary. For example, there was a very nice paper recently out of Schrodinger where they looked at residence times and they introduced the idea of two-step exploration and exploitation, and ideas of that exploration can be integrated into what I just talked about to reduce the number of iterations that you will possibly need to do here. So for the second application, I'd like to talk about another very interesting molecule, which is RNA, and it's becoming a very attractive therapeutic target. So people actually did not know much about this molecule before, simply because it was very hard to characterize experimentally. But nowadays, people are making progress there, and it offers opportunities for novel therapeutics. So there are different kinds of so RNA performs different function, and we can try to develop drugs that target both protein coding and non-coding RNAs, which would be very, very useful, especially in the cases of undruggable proteins. So for this project, we worked with a group at the National Cancer Institute at the NIH. So we looked at this riboswitch, which is considered to be a model system for studying RNA ligand interaction. So this regulates metabolic pathway in bacteria by sensing a specific ligand concentration. And it's also interesting to study because it has highly conserved ligandable pockets, which you sometimes don't get in RNAs. So for this specific system, there is a hypothesis that ligand on rate is actually what determines riboswitch-mediated transcription activation. So K-on is relevant. So in previous study for the protein ligand, it was K-off. Here it's K-on. And our experimental collaborators asked us how to achieve a mechanistic understanding of the interaction. And so the first ligand that you see, that's the natural ligand, and they developed a library of synthetic ligands, and they observed different behavior between these two classes. So they wanted to know how to distinguish these two classes. And but the problem is it's very, very difficult to get on rates out of all out of molecular dynamics simulations. So it's even harder than getting chaos. So what's our approach? So here we get experimental affinity from ITC measurements. And then we do enhanced sampling to observe dissociation events and calculate off rates. And then we can calculate a hybrid on rate by combining these two values. So one other thing is that here, the problem is a little different again. Here we have one target and have a library of ligands. So we also thought about, can we train just one machine learning model and transfer that across the other candidate ligands so that we don't have to train machine learning model every single time. Um, um, so in terms of getting interaction mechanisms, these are some of the results. So we found that water and ion plays a very important role in dissociation. So you can see in the bound state, the ligand is the magnesium. There's a magnesium ion which interacts with 3 water molecules. So before dissociation can happen, a 4th water molecule comes in, which leads to a loss of interaction with the ligand, and then the ligand can exit. We also observed that the RNA itself undergoes a change. So this part, the top part of the RNA is known as a P4 domain, which kind of expands. It's like the mouth of a snake opening, which allows the ligand to exit the cavity. And we also observed that there are differences between natural ligand and synthetic ligands. For example, the synthetic ones are capable of forming pi-pi stacking interaction, as you can see in the top row, and the natural ligand cannot do that. So that also leads to difference in behavior. We were also able to propose ligands with different moieties, and we thought that, okay, in the bound state where the distance is less than 0.3 nanometer in that range, can we have multiple metastable states? So we added an extra group, and we saw that you actually can form different alternating metastable states by forming different hydrogen bonds, so which was also very interesting. So finally, we did all of this, and we were able to calculate the hybrid kaon that I just talked about, and we saw different behavior between the synthetic and the natural groups. So to establish the validity of the framework, we actually did for a different ligand. So we trained a machine learning model for another ligand and did everything, transferred the models, and we observed similar trends. And the p-values associated with the Kolmogorov associated with the KS test, you can see it's pretty reasonable. So this brings me to the last part of my talk, which is how can we extract more information or interaction mechanisms out of models, and how the models that we use, and how can we ensure transparency in our model. So this relates to explainable or interpretable AI methods that people have been working on, and some work has been done. For example, the one that I cite over here. So here our motivation is a little different. So what we are thinking is that the trained models that we are using to do the enhanced sampling already has hidden information about the interaction mechanisms, right? Otherwise, the enhanced sampling would not have worked. So can we get that information out of the machine learning models? But the problem is this extraction is not very easy because we cannot look at the entire trajectories to derive mechanism. So I show an example, low dimensional space learned by our model where a ligand dissociates out of the RNA that I just showed. The problem is in different basins or different metastable states, different features are relevant. So there are different mechanisms that are happening in different basins. So if you try to analyze your data or trajectory without considering that, what you will end up doing is averaging out all the feature importance or interaction mechanisms. So what you will learn will not be very useful. So here our idea is that we developed a method which was published last year. So it's a generic model agnostic method that can use on top of any machine learning model. So we developed SPIB for analyzing molecular simulations. So our method for understanding what's going on is in machine learning models is called TERP. So it sits on top of SPIB as a post-hoc interpretation scheme, and it tries to understand which features were relevant for at the transition state, which can tell us about interaction mechanisms. So the idea is very simple. So you can imagine this schematic represents the low dimensional space separating two classes, two metastable states. What this method does, it zooms in at a local neighborhood and draws a straight line as a function of the input features. So because we are building a linear model, you can simply take the coefficients of the linear model and use that as feature importance, and that will tell you something about interaction mechanisms. So application of this method. So this is a paper. This is a work that we have been working with Marcus Seliger at Stony Brook, where we apply TERP to identify relevant features for identifying the for the rate-limiting transition state of drug dissociation for Abl kinase and imatinib. So we train our machine learning model using various different features. So a brief list of all of them I highlight here. And using our method, we identified the most relevant features during transition at the transition state, which I list at the bottom over there. So our experimental collaborators went and did some measurements. So what they did was they mutated the Abl1, some of the Abl1 residues that form contacts with the imatinib, which tells us about the role that those residues play towards affinity and dissociation. So it turns out that what they found in their experiments agree well with what TARP identifies. So you can see the low-dimensional representation out of SPIB in those figures, and then because SPIB also has been shown to identify the commuter, we can go look at the transition state ensemble and ask which features are relevant at this transition state. Because we have to be careful, the relevant features could be different if we fall into different bases. So I think that's why it performed well. So ongoing work is that most of the current approaches, including TERP and some of the other ones, try to approximate behavior of the trained model locally. But one of the challenges, although they can be successful, is that they can be difficult to achieve for complicated systems and they can be very expensive. So what we are working on is to design inherently interpretable variational autoencoders for molecular simulation. So here the idea is the machine learning model is trained to predict which features are relevant on the fly in addition to performing the main task. So multitask learning, and we are hoping to archive this work quite soon. So in conclusion, I tried to give you an overall idea of how we are doing enhanced sampling to observe interesting events. And I showed you applications for both RNAs and proteins. I also talked about how we can harness information about interaction mechanisms and feature importance out of your trained model. So yes, I'm a fifth-year PhD candidate in Pratyush Tiwari's group, and I'm hoping to graduate very soon. So I'd like to thank all of my experimental and computational collaborators for helping me with everything. Yeah, thanks all for your attention, and I'll take questions. Thank you. No, the information bottleneck doesn't relate to the commuter. So information bottleneck is a principle, but after we have imposed some conditions, such as I showed, hold on. So yeah, so if we impose the first principle that I show on an information bottleneck, which is if a system is in a metastable state, it has the highest probability of staying in that state after a small time lag. So we have to mathematically formulate this statement. Once we plug this into our information bottleneck, then we can show that the low dimensional space that you learn, it approximates the commuter, yeah.